This video is going to focus on our humoral response, which is one of the components of our specific immune response. So in our last video, we talked about the cellular response, okay? And I mentioned that that's where T cells are involved. And once a virgin T cell recognizes and binds to a dendritic cell, which is an antigen presenting cell, it can differentiate into three types of T cells: the cytotoxic T cell, the memory T cell, and a T helper cell. Well, this video is where we're going to be focusing on our B cells with our humoral response, but you're also going to see that T helper cell come into play, because even though these are two branches that make up our specific immunity, they are connected to each other. Okay, and T helper cells are kind of going to be that middleman there. So with the humoral response, we're going to be focusing on B cells, which are antigen presenting cells. So that means the B cell is capable of recognizing a pathogen, intact pathogen. It will catch it, ingest it, digest it. Remember, that all means phagocytosis. And then it will present it as well. Okay, so the ultimate goal of the humoral response is going to be to release antibodies. Okay, and so what's going to have to happen is a B cell is going to have to recognize, phagocytose, and present the pathogen, and then a T helper cell is going to come into play. Their recognition of each other will activate or will allow that B cell to differentiate into plasma cells, which can produce those antibodies. Okay, now I just gave you a ton of information in like 30 seconds, so don't obsess over that just yet. All right, we're going to get to worry about detail in a second. But for now, I just kind of want you to see that this is where B cells and those T helper cells are going to come into play. Okay, and just like we saw T cells differentiating into the cytotoxic T cell, the helper, and memory cells, B cells do the same thing, but they're going to be differentiating into memory cells, again, so we don't forget the pathogen, but also plasma cells. And those are the specific cells that release antibodies that are going to go, and round up those pathogens. Okay, so our humoral response, these B cells, are going to be for targeting extracellular pathogens. So if I were you, I would pause this video and I would go back and I would kind of start comparing and contrasting the B cells and T cells, okay? And they're going to have a lot in the contrast column, okay? What they have in common is they're trying to, right, clear up an infection, but they have different targets, they recognize different things. It's going to be really important that you can separate the humoral from the cellular response, which means you're separating what B cells are doing and T cells are doing, okay? So let's go ahead and take a look at the complex process of activating these B cells, and because that's going to be what it takes to eventually allow them to differentiate into the plasma cells and the memory cells. Well, actually, real quick, what I want to show you is this is a B cell, okay, that has not differentiated yet. And notice it's going to have little antibodies on its surface. Here in a couple of videos, you'll know that antibodies have this Y shape. And so it is recognizing this intact pathogen, okay? And eventually, it will become activated and differentiate into this plasma cell. And you know it's a plasma cell because it's releasing all of these antibodies, okay? And what's great about these images is, even though right now you're like, how would I have known this is an antibody? Once you're done watching all the videos for this section, you will, and you'll actually know why they're IgM, which is a type of antibody. Okay, so always make sure, y'all, with this unit that you're paying close attention to the pictures, especially once you've learned all the content. Come back and you'll start to see how things are connecting. All right, so Notice here we have two different images, and that's because in order to activate a B cell, which will eventually allow it to differentiate into those plasma and memory cells, there's kind of like a checks and balances system. Okay, so one pathway is going to be B cells doing their job. The other pathway is getting those T helper cells made. Now, we talked about this process in that last video. Okay, and so that's why we have two things happening here. So I'm going to start with the B cell process, and then I'm going to stop about halfway. And then we're going to bring in our T cells. We're going to show how they interact, and then we'll continue our focus on the B cell process. Because remember, B cells are the main player here for our humoral response. All right, so up here at the top, remember, B cells can recognize intact pathogen. So that's what's happening here. It's recognize the antigen on that pathogen, because notice the entire pathogen is this little asterisk shape, but it's only recognizing one little part that has bound to the receptor. B cells are antigen presenting cells. So that antigen bound, notice here endocytosis, which is the beginning of phagocytosis. And it doesn't show the entire process, but here it has been phagocytosed. And what actually is going to be happening, and kind of a limitation to this image, is right here, the little antigen is on the surface. And you can tell there's this black ring. The reason I call this a limitation is I would have preferred them, instead of having the antigen be a black ring, it should have been that little yellow kind of square region. But the point is, notice it's now presenting antigen. And so this B cell is now considered activated. It says, heyo, look what I found. I have this antigen. That means I caught a pathogen, and there's probably more of them running around in this body, and we need to get them, okay? But remember, I said there's a checks and balances system. Because what if that B cell was wrong? What if it was just a wonky B cell and it found something it shouldn't have, right? We don't want it to just be able to activate the entire immune response and essentially be attacking ourselves. Okay, that could go very poorly. So that's why there's two cells involved, just like with our humoral response, right? The dendritic cell presented the antigen, the T cell confirmed it. So it's kind of the same thing. The B cell is presenting it, then the T cell is going to come and confirm it. Okay, so real quick from the top, the B cell detected the entire pathogen. It went ahead and phagocytosed it, and it's presenting part of that pathogen to the immune system. But now it needs to be checked, which is what's happening over here. Now, but who's doing the checking? Well, this time it's a T helper cell that is helping out here, okay? So let's review where that T helper cell came from to begin with. So remember, from the last video, there was a different antigen presenting cell, a dendritic cell, that caught a pathogen and it presented it. Remember, the naive T cell here bound to it and allowed that T cell to differentiate into T helper cells and cytotoxic T cells and memory T cells, okay? Well, now we're focusing on these T helper cells down here, okay, and memory T cells. Just a reminder there that they're important. So this T helper cell, what's going to happen next is this little T helper cell is going to come over here to this B cell and be like, so you think you caught a pathogen? Let's make sure of it, okay? And so you'll notice they're binding. When there's binding involved, that means there is a match. This little T helper cell has confirmed that yes, the B cell detected and eliminated a pathogen. So they need to get ready because there's probably more pathogens swarming around. Okay, so if I were you, I would mark in your notes that this cell right here is a T helper cell. Okay, and this binding is going to release cytokines from that T helper cell, And it is that release of cytokines that's going to allow that activated B cell to differentiate. Okay, so now notice the B cell looks different because it's differentiated into a plasma cell, which can then continue to divide, make lots of other plasma cells, all of which release antibodies. That is the end goal of this system, release antibodies. Okay, there's a lot happening here, y'all. So this is one of those slides and videos that I would pause and kind of sketch it out on your own as we talk about it, because there's just a lot happening. Okay, so remember, two separate pathways have to come together. Pathway one is the B cell detecting and presenting the antigen. Okay, and it says, hey, I have a problem. The second pathway is making these naive T helper cells. Okay, once the activated T helper cell is there, it's going to come on over and it is going to confirm what the B cell is saying. Once the T helper cell confirms that the B cell did in fact find a pathogen, that's going to allow the T cell to release cytokines, which will cause that B cell to differentiate into antibody producing cells, which are known as plasma cells. Okay, so again, I would recommend going ahead and writing out this process more in like a stepwise fashion. Don't just copy down what's on this slide, but go through and say, okay, what is phagocytosis, right? And include it in these middle steps. Draw in where the T cell pathway comes into play and what the end result is. All right, y'all, that's the end of this first video. The next video is going to start to focus on antibodies, their structure, then we're going to start moving into what they do and how they work. Talk to you soon. Let me know if you have any questions.